and then now we do
Bacterial.
bacteriology. After that, mycology, and then
Virology.
virology by all the germs, and then after that,
Infectious disease.
yes, infection by system, right? So one good thing about microbiology is that it's like a repetition. When you do infection by system, it's like a repetition of all the microbes that you saw, different germs, and also the immunology part. So it's like a combination. So very so that's the fortunate thing about microbiology. It's like re-emphasizing in a way, and also looking at it in a different way, by system, by infection by system. Okay, so today is our first lecture on bacteriology. So bacterial, cell and genetics. Before I start, is everyone in? Yang depan memang always empty. Okay, so before I begin, when I say bacteria, what bacteria comes to your mind? Any specific bacteria that comes to your mind? Yes? Like here, E. coli, lagi?
Streptococcus pyogenes.
Streptococcus. Yeah? Salmonella.
Neisseria.
Yes? Any more? Enterobacter. Any more? Helicobacter. Mycobacteria. Neisseria. Okay, very good. None of you have given me a viral name or fungal name. So you know, bacteria, although sometimes you might get confused, but it's the sooner you get familiarized with the names, the better for you, because you'll be seeing this over and over. And then sometimes at the end of the course, students feedback selalu ada banyak sangat ingat, too many microbes to remember. Of course lah, this is microbiology and you are going to be a doctor. Memang you have loads of information that you need to get into your head, not only microbiology but other subjects. So always repetition and recalling is very important from very beginning. Okay, for your name, to make sure you remember all the information, and then they form a basis for your clinical years as well, right? So don't always must have a eukaryotes. Virus pro. What are viruses? Any answer?
COVID-19.
They are neither, right? Neither pro nor eukaryotes. They are non-living cells, right? They need a host cell to show living being characteristics, right? So they are not classified as eukaryotes or prokaryotes. Okay? So in fact, you don't call a virus a cell. When you're studying virology, when you see cell, they are meaning the host cell. Okay, so viruses, they are not even cells, they're particles, viral particles. Okay, so first of all, we want to differentiate between eukaryotes, prokaryotes, and then we have all the structures of bacteria, and then how we stain them to get to identify the bacteria, and then some important information on bacterial genetics. Okay, so let's go through this very quickly. So first of all, prokaryote versus eukaryote. Pro means before, eu means true lah. So they are what is it that is true here and almost true here is the karyote. And what is karyote? Karyote actually means
Next is
Karyos comes from the word karyos, means kernel. Kernel. You know what this is? Yeah? Kelapa sawit. Yeah, okay. So, Malaysia kan, so I put kelapa sawit lah. Okay, so kernel is the center part, right? So nucleus, the word karyos came from kernel lah. The meaning is actually kernel. So kernel is the central circular part. Okay, so prokaryote is referring to the nucleus lah. Okay, the nucleus. So what is different in the nucleus of eukaryotes and prokaryotes is that the nucleus in prokaryotes are not even called nucleus lah, okay, because they don't have the membrane. So without membrane, it's called nucleoid. Okay, so this is a nucleoid. So it contains the genetic information like a nucleus, but it doesn't have membrane. That's all. Whereas true eukaryotes, they have nucleus. Okay, so this chart is roughly to tell you where the bacteria sits in a chart if you want to differentiate between the sizes lah. Okay, so this is in a micrometer scale. Why I cannot, the mouse doesn't work. My pointer. Okay, so anyways, if you can see, the first is the polio virus. Polio virus is the smallest virus, and then pox virus is the largest virus. So you can see pox is in somewhere in the middle, and then you see hepatitis B virus, HIV. Okay, and then you see mycoplasma. Mycoplasma is the smallest of bacteria lah. Clinically important one, mycoplasma. And then you have Haemophilus influenzae somewhere there. Escherichia coli about 1 micrometer. Bacillus anthracis about less than 3. Candida, what is Candida albicans? It's a fungi, yeast. So that's why in the and then the red blood cell is larger than all these. Okay, so and then comes your protozoa lah. That's why when you're looking under the microscope for bacteria, you have to look up to 100 times. You put the oil immersion to make it more clear lah, bigger to see clearly. Whereas fungi, you can see at 40 times. You can already see the fungal cell. Okay, and viruses, of course, you cannot see using our usual microscope in the light microscope in the lab. You need the electron microscopy and so on. Okay, so that's just a rough idea to see where our bacteria sits in. Okay, so these are the structures of bacteria. The differences between eukaryotes and prokaryotes. So most others are eukaryotes except virus, and then we have the bacteria. Size is about 0.5 to 3 micrometers. As I said earlier, no membrane, nuclear membrane. Okay, cell membrane adalah. Okay, chromosomes. Eukaryotes have strands of DNA diploid genome. Okay, as the chromosome, whereas prokaryote it is single and circular. And haploid genome. Okay, and cytoplasmic structures within the cytoplasm. There's mitochondria present in eukaryotes, which are absent in prokaryotes. Golgi bodies absent in prokaryotes. Endoplasmic reticulum absent. So mostly all the nuclear particles are absent. Okay, membrane bound particles are all absent in the prokaryote. Okay, and no mitochondria as well. Ribosomes, there are two types. Okay, so eukaryotes have the 80S ribosome, prokaryotes 70S. Okay, Cytoplasmic membrane contains sterols in eukaryotes. Okay, like humans we have cholesterol, right? So eukaryotes have sterol, whereas bacteria they don't, except for mycoplasma. Okay, it contains sterols. Cell wall. Okay, besides the membrane, bacteria has an additional cell wall. In eukaryotes, fungi have an additional cell wall that is thick. Okay, and but prokaryotes, eukaryotes generally like humans, animals, don't have this extra cell wall lah. Okay, so this cell wall is a complex structure containing protein, lipids, and peptidoglycans in the bacterial cell. Okay, reproduction in eukaryotes is sexual. In fungi there is sexual and asexual, right? In prokaryotes there is only asexual, which is binary fission. Okay, binary fission meaning dividing lah, one cell becomes two. Okay, movement. In prokaryotes they have a flagellum for their movement, but not all bacteria have flagellum. That is why not all bacteria are motile. You will see in bacterial identification in the lab that one of the criteria that you look for in bacterial identification, especially in gram-negative bacteria, is their motility. Okay, by looking at its growth on in agar medium. Okay, so the motility is important. So when they have flagella, they are motile lah, can move. Okay, respiration. Here we have mitochondria. This cytoplasmic membrane. Okay, I want to go through this very quickly so that I don't rush in the end. So we have the cytoplasm that contains all those I mentioned earlier, right? Cytoplasm is about the same lah. Okay, 80% water and all the nutrients. Okay, and then has the nucleoid inside it. Okay, nucleoid, as I said earlier, is a single circular molecule with no membrane. Plasmids and transposomes we'll see at the end of the lecture later. Ribosomes. So ribosomes is the site of protein synthesis. So one of the reasons we want to know the difference and also characteristics of bacteria is mainly because one, we want to understand their pathogenicity, etc., but also because we want to, in order to get rid of them, in order to kill them, you must know lah, okay, what is different in them, so that you can target. Okay, antibiotic drugs can target that structure without harming the host structure. Okay, so these differences are important for that. As you can see later, some drugs target the ribosome, some drugs target the cell wall, because we don't have those that type. Okay, nutrient and storage granule. Okay, so these are granules stored in some bacteria. I think only one that you would come across during this course, if I'm not mistaken, will be Corynebacterium diphtheriae, right? Of course, Mycobacterium important, but we don't look for this in the lab lah. Okay, but the metachromatic granules are present in the Corynebacterium diphtheriae. This is an important feature to differentiate it from other bacteria, gram-positive bacteria. Okay, so it's a storage granule, and then we go to cell envelope. So the cell envelope in bacteria has membrane and wall. Okay, so the membrane is the usual phospholipid bilayer. Okay, and then it has only mycoplasma has sterols. Okay, the usual phospholipid bilayer. So what it does: active transport of molecules into the cell, energy generation, okay, synthesis of precursors of cell wall, and secretion of enzymes and toxins. Okay, so this is very important for us to understand the difference between gram-positive and gram-negative cell wall structure. Okay. So what is gram, by the way? Gram is the scientist's name lah. Okay, it's just the name of the scientist who created this staining technique. Okay, gram stain is a staining technique to differentiate these two groups of bacteria, two large groups of bacteria. When specimens from patients are sent to the microbiology lab, to the bacteriology unit. Okay, one of the first tests that is done on the specimen is to a gram stain. Okay, the specimen is taken and swabbed on a slide. At the same time, it's grown on agar plate and all kind, but that will take time to grow. The first most method that can help differentiate between bacteria until you identify the species or strain or genus of the bacteria that is causing the infection. Okay, the very first step would be to differentiate it by gram stain. Okay, so generally there's gram positive and gram negative bacteria. Okay, so the difference between gram positive and gram negative, what can you see? What's the difference? Yes? Yes? Can't hear. The peptidoglycan layer, the outer membrane, yes. So peptidoglycan layer is thicker, right, in gram positive. Outer membrane is not present in gram positive. It's only there in gram negative, right? That's very important. This is a pointer. Okay, so this is the peptidoglycan layer, right? And then this is the outer membrane, right? Okay, we'll come to that. So now we only see then what else? Oh, if you look at this here, this is gram positive, gram negative. Gram negative has a periplasm or periplasmic space in between. Okay, periplasmic space in between the membrane and the outer membrane. There is one space. It's called a periplasmic space. Okay, so peptidoglycan is composed of a sugar backbone, glycan with peptide side chain. So it's a very complex structure, as you can see here. Right, so it has the NAM, N-acetylmuramic acid, and N-acetyl glucosamine molecules. Okay, and then each muramic acid molecule there's a tetrapeptide, and then there is a peptide chain cross-linking. Okay, so this structure is this complex structure helps the structure of the whole structure of the bacterial cell lah. Okay, the integrity, okay, to make sure it doesn't burst osmosis and so on. Okay, so it keeps the bacterial cell intact because bacteria is just a single cell kan, so it needs this feature to keep it strong lah, okay, in the environment. Okay, it's a similar picture. Okay, I'll just say this again. The link is not working. The mouse is not working.
Osmosis. The synthesis of the peptidoglycan and ultimately the bacterial cell wall occurs in a number of stages. One of the first stages is the addition of 5 amino acids to N-acetylmuramic acid. Next, N-acetylglucosamine is added to the N-acetylmuramic acid to form a precursor of peptidoglycan. This peptidoglycan precursor is then transported across the cell membrane to a cell wall acceptor in the periplasm. Once in the periplasm, the peptidoglycan precursors bind to cell wall acceptors and undergo extensive cross-linking. Two major enzymes are involved in cross-linking: transpeptidase and D-alanine carboxypeptidase. These enzymes are also known as penicillin-binding proteins because of their ability to bind to penicillins and cephalosporins. Eventually, several layers of peptidoglycan are formed, all of which are cross-linked to create the cell wall. Gram-positive bacteria may have more layers than gram-negative bacteria, and thus have a much thicker cell wall. Beta-lactam antibiotics include all penicillins and cephalosporins that contain a chemical structure called a beta-lactam ring. This structure is capable of binding to the enzymes that cross-link peptidoglycan. Beta-lactams interfere with cross-linking by binding to transpeptidase and D-alanine carboxypeptidase enzymes, thus preventing bacterial cell wall synthesis. By inhibiting cell wall synthesis, the bacterial cell is damaged. Gram-positive bacteria have a high internal osmotic pressure. Without a normal rigid cell wall, these cells burst when subjected to the low osmotic pressure of their surrounding environment. As well, the antibiotic penicillin-binding protein complex stimulates the release of autolysins that are capable of digesting the existing cell wall. Beta-lactam antibiotics are therefore considered bactericidal agents. Bacterial resistance to beta-lactam antibiotics may be acquired by several routes. One of the most important mechanisms is through a
you can watch this related to the bacterial genetics. Okay, so from the video, and there's one part where it talks about transpeptidase, the one that is like peptide, cross-linking peptides, right? Cross-linking peptides. So this transpeptidase, another name for it is penicillin binding protein, okay, because penicillin targets this transpeptidase, okay. Penicillin binds to this transpeptidase and inhibits it from doing what it's supposed to do, okay, to form the cross-link. So the bacterial cell wall, yes? Oh, sorry. Okay, so it inhibits the transpeptidase from doing what it's supposed to do, put the cross links, okay, to make the cell wall more rigid. So when there's these cross links are not formed, the cell wall is not strong and it can lyse. Okay, so that's a way of killing the bacteria. Okay, penicillin. Okay, this one also wordy. So this is gram stain. What is gram stain? Okay, so basically gram stain has got 4 reagents to it. Okay, number 1. But before you do the gram stain, first what do we do? You will be doing this in your first bacteriology lab, which is after your break, right? After your break. Okay, so before you stain bacteria, whether it's gram stain or any other staining technique, first you must make a smear. Okay, can you see this? Yeah. You have not done this yet? Okay, so this is a slide, microscope slide. The other day you all took your blood culture, drop of blood, you put on the slide, right? Okay, so this is a microscope slide. So what we do is we pick the bacterial colony. Okay, so you pick the colony meaning the bacteria that's growing on its media lah. Okay, you can also take from specimen. Okay, as I said earlier, even from specimen you can directly make a smear, but it's clearer when you do it from the colony lah, because you're taking pure colonies. Okay, so it definitely has one type of bacteria. Okay, but in the hospital, for some specimens they do direct from specimen. Okay, so a smear is made. You will be explained more thoroughly on the during lab. Okay, and then after the smear is made, the first reagent added is crystal violet. Okay, crystal violet, it gives the first stain. What color is it?
Purple.
Purple. And then iodine is added. Okay, iodine acts as a mordant that makes a complex with the crystal violet to make sure the color is steadfast lah. Okay, so this color will go into the bacterioglycan layer. Okay, colors the cell wall, right? And then we add the alcohol wash. So alcohol will decolorize only in gram negative because it's easily decolorized lah. Okay, because it has a thin bacterioglycan layer, easily decolorized. Okay, but it has to be done very quickly. If you decolorize very long, if you add lot of alcohol, even gram-positive bacteria pun will hilang lah the alcohol. Okay, so this technique it has the timing lah for it. Okay, so it's decolorized, and then you add safranin, the counter stain. Okay, you call it a counter stain because it's the next stain lah. Okay, you add the counter stain. If it has been, the bacteria that are decolorized will now take up the counter stain, which is what color? Pink. Okay, so as you can see here, there are two types of bacteria. Okay, gram-positive bacteria, it doesn't lose the color purple. Okay, it retains purple till the end. The gram-negative bacteria, the color is removed after alcohol, so it becomes pink. Okay, so it's just very simple. So gram-negative bacteria are pink, gram-positive bacteria are purple. But when you look under the microscope, you are not supposed to say pink or purple. You must say gram-negative or gram-positive lah. Okay, even for your speed and the end of the semester and so on. Okay, so important to remember that. So this is a gram stain of Escherichia coli, E. coli. Okay, and this is a gram stain of Staphylococcus aureus. Two examples of gram-negative and gram-positive bacteria. Okay, bacterial shapes. So bacteria has got shapes. Okay, as you can see here they showed negative, circular and rods, right? But not all gram-positive are circular and not all gram-negative are rods. This is just an example. Okay, so bacterial shapes, the main ones are coccus and bacillus, and then we also have spiral shaped. Okay, some bacteria are not very common. Coccobacillus is bacillus shape with a rounded edge, so it looks a bit like cocci. Okay, so it's called coccobacillus. And then you have the comma shaped bacteria, which is only one, which is Vibrio cholera, which causes cholera. Okay, only that is comma shape. Okay, this one is just a joke. Looking like coccus. Bacteria doesn't grow in size so much. Okay, so this is just a joke. Okay, the bacteria grow in number. They don't grow in size. They multiply. Okay, some examples of coccus and then the arrangement. Okay, so Streptococcus pneumoniae is a diplococci. It's present in pairs. Okay, another diplococci is Neisseria gonorrhoeae. Okay, which is a gram-negative bacteria. It's not pink because it's gram-negative. This is electron microscopy, so the color doesn't matter here. It's in our light microscope that we see the gram stain. Right? Okay, so this one they use different color enhance lah. Okay, and then cocci arranged in chains. One example is Streptococcus pyogenes. Okay, that word strep itself because it's like a chain. Okay, streptos. Okay, this is grape-like cluster. Okay, but in reality, you won't see like this in the microscope. They will be like more like this type of background lah. Okay, so this is a micrococcus with tetrad arrangement. This is cocci in cube. Sarcina.
Sakina.
We won't see that also. Okay? And then these are Pseudomonas aeruginosa. These are bacillus. Okay, bacillus we don't. We either call them bacillus or rods. Okay, and they are usually in singles or very seldom we see chains. Okay, usually in singles. Okay, example is Pseudomonas aeruginosa here. A cocobacillus that you would be coming across is Haemophilus influenzae. This is a bacteria, not virus. Don't confuse with influenza virus. This is Haemophilus influenzae. And then fusiform is another form of rod bacteria, shaped bacteria. So it has the edge is like a sharp needle. Example is Fusobacterium nucleatum, usually the dental microbiology lah. This one. Okay, and then spiral shape, Vibrio, Spirillum, Helicobacter pylori is also spiral shaped. Okay, and it has a flagellum tuft on the top. It's called a helicopter. Okay, and then some bacteria are very thin walled and they are spiral shaped like Leptospira interrogans. That causes leptospirosis, right? And they are very very thin, so it's very difficult to gram stain them. Okay, so it's you will see that lah in the lecture. Okay, this is another spirochete, Boridia. Okay, so now just showing you this bag because just now I showed you for the color, the gram stain color. Now can you see that these are, what shape are these? Rods, as compared to these are cocci, okay, or coccus. So when you are describing them under the microscope, when you describe them in your question on your notebook or your exam sheet or whatever, you call them gram negative bacilli. Okay, these are gram positive cocci in clusters because they have a special arrangement. They are in clusters. Okay. Okay, as compared to this one here is also gram positive cocci in chain. Okay, it's not in cluster. Although over here you might see cluster, but if you see chain in most part of it, then it's a chain lah. Okay. This one is a typical Staphylococcus. You will never see a chain in it. Okay, it's even if you see, it's actually a cluster. Okay, very short maybe the chain. Okay, so this is a rod, gram negative rod. This is a rod. Okay, and this one is not gram stain. This is acid fast bacilli. Okay, you will do this when you come to it lah. But you can read up your SLP on this. Okay, on basic diagnostic techniques in bacteriology. Okay, it cannot be gram stain. You know what organism is that? Mycobacterium. How do you know? You have studied in pathology. There. Okay? Have you shown this?
Macam tak.
Yes? Yes, you've been studying hard.
Tadi
How do you know this mycobacterium?
dia
Mycobacterium is the blue ones or the red ones?
blue.
The red rods here, they are arranged in like circular serpentine arrangement. Very very thin rods, but can't really see clearly. I can see clearer on this screen here lah. Okay, there's a reason why they are called acid fast. I don't want to go into that now yet. Okay, so back to that. Next, we have the outer membrane and the
Immune.
lipopolysaccharide. Okay, so this is a very important component in which bacteria? Gram-negative bacteria. Okay, remember earlier that the lipopolysaccharide is only present in gram-negative bacteria. Another name for lipopolysaccharide is endotoxin. Endotoxin. So only gram-negative bacteria produces endotoxin. Okay, so the components of a lipopolysaccharide is the toxic lipid A. Okay, the lipid A part and the bottom, right, and then the core polysaccharide and the O antigen. Okay, so the O antigen has got all the different types of sugars, so it's also used to identify gram-negative bacterial strains using serotyping. Okay, and the toxic lipid A is the toxic part that gives its name endotoxin. Okay. Then some bacteria also have teichoic and lipoteichoic acids. Okay. So you can see the teichoic and lipoteichoic acids here. So mostly present in gram-positive bacteria, but also the gram-negative. This is the periplasmic space I was talking about. Space between the inner and outer membranes in the gram-negative cell wall. There's a space which contains peptidoglycan layer, penicillin binding proteins, and hydrolytic enzymes. Okay. Including beta-lactamase. Okay, so beta-lactamase, it can degrade beta-lactam like penicillins. Okay, so all these enzymes are present there. So generally, back to the difference between gram-positive and gram-negative cell wall. So gram-positive has got no lipopolysaccharide. Gram-negative either lipopolysaccharide. Gram-positive peptidoglycan is thicker. Gram-negative thinner. Periplasmic space only present in gram-negative bacteria, and teichoic acid often present in gram-positive. So this is the summary of the difference between gram-positive and gram-negative bacteria. Okay. Next, we move to the external structures. For example, the capsule, flagella, membrane, and glycocalyx. Okay, so the capsule, not all bacteria has this. So it's a gelatinous layer covering the bacterium, and it's composed of polysaccharide. Okay, so except for Bacillus anthracis that causes anthrax. Okay, for that it's not a polysaccharide, it's a polymerized D-glutamic acid. And the sugar components also varies. For example, like Streptococcus pneumoniae, it has more than 80 serotypes. Okay, so based on that is characterized based on the capsular antigen. Okay, so even the types of vaccine is dependent on this, targeting bacteria with a certain number of capsular polysaccharide. Okay, Streptococcus pneumoniae. Okay, so why is it important? It is a virulence determinant. Okay, it can prevent phagocytosis. So capsule makes it more difficult for the immune cells to capture the bacteria. Okay, so it prevents phagocytosis. It's used for identification using anti-serum against the capsular polysaccharide. So this test is called the Quellung reaction. Okay, and then the capsular polysaccharides are used as antigens in vaccines. Okay, because they are very immunogenic, right? And they can adhere. It helps them adhere to the host. They stick to the host. This capsule. Not all bacteria have this. Usually, bacteria that have capsule When you grow on agar plate, they will have like a watery, like berair, yeah? Watery kind of morphology. Okay, like here, right? So this is bacteria growing on the agar plate. So as you can see, it looks like. Okay, so as you can see, it has a kind of like mucoid appearance, like a watery appearance. So this is a negative stain of Streptococcus pneumoniae. You can see it's polysaccharide there. Okay, negative stain means what? Not gram negative, eh? What does negative staining mean? You stain the background. You don't stain the organism, you stain the background. Okay, so slime layer. Okay, so this is also polysaccharide coating secreted by many bacteria, and it gives firm adherence to various structure including plastic materials. Okay, it is a component of biofilm. I think we will look at biofilm when we do bacterial pathogenicity. Okay, so it can stick not only to the whole cell but also in the environment in plastic materials. Okay, and devices, plastic devices in medical devices. So it's a very important virulent mechanism for bacteria forming biofilm. Flagella, as I said earlier, it's for motility. So many rod-shaped bacteria have got flagella. It's it's not okay. Okay, so you can see the flagella at the end of the bacteria, kan? To help it, it's like whip like appendage. Okay, whip. What do you say, whip? Yeah, Malay, what do you call whip? Okay, these are the different types of flagella. Okay, that's based on the structure. There's monotrichous, for example, in Vibrio cholerae. Lophotrichus, there's a few flagella attached at one end. Okay, that's Lophotrichus, like the Bartonella. Amphitrichus, both ends. Peritrichus all around, like in E. coli. Okay, Escherichia coli has peritrichus flagella. So Escherichia coli is a very important urinary tract infection causing bacteria. Okay, and the source of it is usually from feces. Okay, usually. Okay, so the ability to be motile helps it to swim up. Okay, and further infect the urinary tract. Okay, the bladder and so on, and to be more virulent compared to other bacteria that can also be there. Okay, so let's explain here. Okay, some species of bacteria are identified by the use of specific antibodies against flagella proteins, just like capsular polysaccharide. Flagella protein are used to, the flagella protein in the bacteria are used to identify the bacteria. Okay. Pili is the tiny hair-like structures you see around this bacteria here. So there's flagella around it, the longer hair-like structure, right? Flagella is very long compared to pili, very tiny. Can you see? Right, tiny around the bacteria. So this is an Escherichia coli, by the way, that is going through binary fission. You can see it's dividing into two. Okay, so pili is mainly found in gram-negative organism, important for adherence to host and in conjugation. We'll see what conjugation is later. Next, bacterial spores. Do all bacteria produce spores? No, huh? What are medically important? There are two genera. What are they? Then you must remember. Two medically important bacteria that produce spores: Bacillus and Clostridium. Okay, Bacillus and Clostridium. Okay, so they spores in bacteria are structure that keeps them dormant in very adverse environment, very cold or very hot environment, very dry environment. They can still survive, and then after even years, if you give them a nice environment to grow, it can germinate and grow. Okay, so that's why some of these bacteria that are present in soil, if the grains from agriculture are taken and for cooking, if it's not well cooked, they can cause food poisoning. Okay, so that's just one example. But even in clinical setting, this is the reason, I think, during your first practical I did mention, right, on why we need to autoclave, why we have to kill the bacteria for 100. We need to heat the pressure cooker, like a pressure cooker kan, autoclave. Why we need to heat it up to 121 degrees Celsius. So you cannot heat water at normal environment, you cannot heat up to 121. That's why we apply pressure. So under pressure only you can heat it up to 121 degrees. So these spores can only be killed at 121 degrees Celsius. Okay, so that's the main reason your autoclave temperature is 121 degrees Celsius under pressure for 15 minutes, right? So these are important facts that you have to remember. Numbers, these numbers. Okay, so they are very resistant to heat, desiccation, freezing, so they can survive. Okay, until it is more the environment is more suitable for growth. Spores or endospores. So there's two, as I said, Bacillus and Clostridium. The species, nothing like when we do gram-positive bacteria. Okay, so this is how the sporulation happens. Not to worry too much. Okay, now we move to bacterial genetics. Is the time? Okay. We do it on time. Okay, bacterial genetics. Okay, so all these important characteristics of bacteria in terms of antimicrobial resistance, virulence, all these are contained within its genetic property lah, right? Some bacteria are more virulent than others, right? Okay, so this is some revision from your biochemistry. I think you might have learned all this kind. DNA replicates, transcribe, RNA, right? RNA forms the base for making protein, right? Translation. Okay, first let's see how bacteria reproduce. Okay, bacteria reproduces through binary fission. Okay, so when it's binary fission, one bacteria becomes 2, 2 becomes 4, and so on. The genetic material is copied, right? Exactly the same. So then how does bacteria get genetic variation? You need to have genetic variation, right? Like diploid cells, like us or eukaryotes. There's always 2 copies of the gene, kan? 2 copies of the DNA, one recessive, one dominant, right? So that gives the variation from parent cells. In bacteria, what gives variation is mutation and genetic transfer and recombination. Okay, so mutation, you know that it is changes in the bases, right? So the shifts or deletion of a base, okay, or substituted with another base or all the GATC kan. So bacteria are haploid, therefore any gene that has mutated is not expressed and results in a cell that has lost the trait. So when a bacteria mutates, the daughter cell would have lost whatever trait lah that the initial bacteria had, right? And main reason, one of the important thing about bacterial variation through all these mechanisms is that it can acquire antimicrobial resistance, right? Bacteria does not vary as much like virus because the replication is very much slower. But virus replicates very fast and it can mutate more rapidly. Okay, and then it gives all kinds of different variants. That's why we have, from time to time, we have emerging more of viral infections that are emerging and dangerous, right, as compared to bacteria. Bacteria pun ada but less, and mostly due to antimicrobial resistance. Okay, over time, because of exposure to antibiotics, okay, some bacteria will acquire the resistance, okay, which makes them more virulent, but very seldom it forms like a new variant lah, like virus kan. Okay, and one of the mechanism is through mutation. Okay, through mutation also bacteria can become more resistant. One good example is the MRSA, which is a very important Staphylococcus aureus bacteria, methicillin resistant Staph aureus, because mutation in that bacteria has caused the MAC-A gene, which is the transpeptidase kind of gene in Staph aureus. So because of its mutation, penicillin does not work for MRSA bacteria. Okay, so that's just you know the importance of it. Next, genetic transfer and recombination. So this is the exchange of genes between two DNA molecules to form new combinations of genes on a chromosome. So bacteria can exchange genetic material within bacterial cell and also between bacterial cell, even between species, right? They can transfer their genetic information. So how? This is just a comic. So from a medical point of view, the most important consequence, as I said, is antibiotic resistance genes. Okay, so it's spread from one bacterium to another. So number one, the first one method by which bacteria can transfer genetic material is through transformation. Okay, transformation. So transformation, there's three mainly: transformation, conjugation and transduction. Okay, in conjugation, bacteria takes up fragments of foreign DNA, okay, and incorporate them into their genome. Okay, so for example, bacteria in the gut, there is this resistant bacteria. For example, when it's dead, the gene can be taken up by other bacteria that are usually not resistant. Okay, they can take up, and then they also can become resistant. This is one of the reasons why when taking antibiotic, they ask to complete the course, right? Because you don't want to, you want to kill the bacteria completely. Okay. I mean, that's a bad example to use here because in transformation, dead bacterial genes are taken up. Okay. So generally, antibiotics have to be finished. Or don't take it at all lah. If you need to take, then complete it. Okay, so this is an experiment where transformation was first described. Okay, so Griffith, Griffith, I don't know, I can't remember now which country. So in 1928, he showed transformation using mouse models. Okay, so what he did was he had the R strain and the S strain of Streptococcus pneumoniae. Okay, so the S strain is the virulent one and R strain is non-virulent one. Okay, so the virulent one has capsule. Virulent means more prominent in causing infection, right? Because at the capsule you can adhere better, also you can avoid phagocytosis, right? So the S strain with capsule was injected into a mouse. Okay, so when injected into mouse, they live, right? So the R strain is the one without capsule. Okay, without capsule. Sorry. Okay, so without capsule, it's not virulent. The mouse is not killed. In the second treatment, the S strain is injected into the mouse. S strain consists of the capsule. You can see it around the bacteria, and the mouse dies. In treatment 3, the S strain that is heat killed, okay, S strain which is already heat killed is injected into the mouse, which means the S strain is no longer alive. Okay, but it's the structure, everything, all the material is there intact, is injected into the mouse and the mouse lives. Okay, so in conclusion, killed S strain cells are benign, they don't cause infection. And in treatment 4, he mixed the R strain, which does not contain capsule but alive, with S strain that contains capsule but dead, heat killed. And he injects this into the mouse and the mouse dies. So this is an example of transformation has occurred. Okay, so live R strain cells were transformed to S strain. So this is an example, the first example of transformation. Okay, next is transduction. Okay, transduction. So this is one way that antimicrobial resistance can be spread between bacteria. Okay. Transduction is a transfer of fragments of DNA from one bacterium to another bacterium by a bacteriophage. You know what is a bacteriophage? Okay, I will answer there. So it's a virus. Bacteriophage are viruses that infect bacteria, but they can also, while they infect, they can also transfer genetic material from the bacteria that they infect to the next bacteria that they infect. Okay, so this is a typical image of a bacteriophage. Okay, so bacteriophages are bacterial viruses. Okay, so they have either a lytic infection or a lysogenic state of living period. Lysogenic infection meaning the bacteriophages infect the bacterial cells, replicate to large numbers, and then they cause the bacterial cell to die. Okay. In lysogenic state, bacteriophages infect bacterial cells and they integrate into the host genome without killing the host. Okay, so that is lysogenic. Okay, so lysogenic conversion can change a non-pathogenic organism into a pathogenic one. Okay, so these toxins, diphtheria, botulinum, cholera, and Streptococcus pyogenes erythrogenic toxin. Okay, so these are all encoded by genes of the integrated bacteriophage. So, for example, like diphtheria toxin, Corynebacterium diphtheriae, if they are not infected by a bacteriophage that carries the toxin, and if it is not get integrated into the bacteria does not will not be causing diphtheria. Okay, it's not pathogenic Corynebacterium. Okay, so these are some examples of virulence factor, the toxins that can be integrated from one bacteria to another by this bacteriophage. Okay. Okay, this is just an animation of the bacteriophage. It infects a bacterial cell, the genome penetrates in, and then it replicates, and then the bacterial genome itself breaks and gets into the, sometimes it is taken up by the bacteriophage, and then when it infects another bacteria, it will transfer the part of the genome, and if that particular genome is carrying antimicrobial resistance gene, then that gets transferred as well, right, to the bacteria. It's a very old animation which I find very useful because very quickly it can show. This is generalized transduction. Specialized transduction is phage DNA that has integrated into bacterial DNA. It is excised and it carries with it at the adjacent part of the bacterial DNA. Okay, so it's a very specific form of transduction. Okay, so when it breaks open, it carries a certain part of the bacterial DNA, but it doesn't kill the bacteria entirely. What does it? When it releases, it does kill. Okay, so this one is specific because a specific gene from the adjacent part is carried. Okay, you don't have to know details of this, more for research and so on, but it's good to know there is specialized and generalized transduction. Okay, so this is a diagram. That was transduction. Okay, transformation takes up dead fragments of DNA from dead bacteria. Transduction, genetic material is transferred by bacteriophage. In conjugation, DNA is transferred from donor to recipient through direct contact. The direct contact is mediated by a pili. Okay, we saw pili tadi kan. I told you one of the functions of pili is to adhere to the host cell, and one more is for conjugation. Okay, so we also mentioned plasmid. Later I'll show what a plasmid is. So plasmid carries genes enabling the bacteria to form a sex pili. Some bacteria, they have this F plasmid form a pili, a certain type of sex pili, meaning it can conjugate. Okay, and in high frequency recombination cell, the bacterium with a conjugative plasmid is integrated into its genomic DNA, and the resistant plasmid is a plasmid that codes for multiple antibiotic resistance. So if the bacteria that is going through conjugation also has a plasmid with antimicrobial resistance and conjugation occurs successfully, it can transfer even the whole plasmid into another bacteria. Okay, so that is conjugation. Okay, this is another. Okay, so we saw transformation, transduction, conjugation. Other important elements of bacterial genetics is the plasmid and transposon. As I said, plasmid, it's actually a circular double-stranded extra chromosomal DNA. Extra chromosomal meaning out of the chromosome okay, additional to the chromosome. There's also DNA stored in plasmid, and it's capable of autonomous replication. It can replicate by itself, not only when the bacterial cell is dividing, it can replicate by itself. Okay, and it's very important in genetic recombination that occurs in bacteria, and it carries genes for all these functions lah, antibiotic resistance, exotoxins, pili, resistance to UV light also. Okay, it can carry those genes. Okay, and I'll show that. Okay. Another one is transposon. Transposons are also called jumping genes. Jumping genes. They are pieces of DNA that can move from one location on the chromosome to another, from plasmid to chromosome or from chromosome to plasmid. So these are genes. They have they are flanked by sequences that can be cut off from the chromosome. Okay, and then they can stick to another part of a plasmid or chromosome in another bacterial cell. So when this happens, and if this jumping gene contains an antimicrobial resistance gene or a virulence gene, then this also gets transferred, right? Including toxin, genes that encode for toxin. Okay, so non-pathogenic bacteria can become a pathogenic one. Okay, so recombinant DNA technology is the technology that scientists use for they manipulate the ability of bacteria to transfer and recombine. Okay, so recombinant DNA technology is it's done in the lab in order to manipulate bacteria and produce certain proteins, for example, by using this bacteria. Okay, so plasmid, this plasmid PBR322 is the most commonly used plasmid from E. coli cloning vector. You know, you've heard of cloning, kan? So this is to clone a bacterial cell into another by especially if you want to produce a certain protein in the lab. For example, the two proteins manufactured, for example, through biotechnology are insulin and human growth hormone, which can be grown in the lab. Okay, insulin for the use of patients. What is done is the human insulin producing gene is recombined into a plasmid, bacterial plasmid, and then it is induced or transfected into a bacterial cell. Okay, so that they become transformed. So you are doing transformation as well here, but not all bacteria can be easily transformed. They are called competent. Only competent cells are transformed. So in the lab, it's given an electric shock or something like, or heat treatment in order for it to take up the plasmid. Okay, so this plasmid, so the bacteria multiplies and then it can ferment and produce insulin lah. Okay, it produces insulin in this way, and then it's purified for use. So that is just a way of manipulating bacterial ability to transform genetic material and to recombine for human use. Okay, other aspects of bacterial genetics. You have all heard of PCR?
Yes.
Okay, so in polymerase chain reaction is used to identify microbes, right? You know how it works. You've done it in biochemistry, I'm sure. So in bacteriology, usually qPCR is not much used, okay, and not in identification lah. Okay, that's more for research and so on. But for viral identification, yes, we use qPCR a lot. For bacterial identification, we do use multiplex PCR for identification, concurrent identification of Neisseria gonorrhea and Chlamydia trachomatis, both to identify sexually transmitted infections kan. So you do a PCR because you want to use the specimen, okay, that for identifying a sexually transmitted disease. So you will do a multiplex PCR where you can determine both, whether the patient has Neisseria gonorrhea or Chlamydia trachomatis. Okay, so that's just an example. Okay, and this is the last slide. Another gene-based method in identifying bacteria is using the 16S ribosomal RNA sequencing method. Okay, so this is because of the difference in our ribosome. Okay, so this is a very powerful method where we can now identify bacteria that is present in a very complex environment, like in the soil. Okay, soil environment, there's a lot of bacteria, and also in our gut in the stool, in the feces, trillions of bacteria. So it is able to characterize the different bacteria present in a environment like that. Okay, and then that in this way we now know that the human microbiota, we call it microbiota, right, in the gut, it's so different between disease and healthy person, healthy people, not only in infectious diseases but even in non-communicable diseases, even in obesity, in diabetic patients, in cancer patients. They see that microbiota that's present in the gut is very different when compared to the normal bacteria, normal patient, normal subjects, humans, okay, with no disease. Okay, so now healthy people are found to have more diverse bacteria in the gut compared to the those who are affected by a disease. Okay, and so there's a lot of research being done on how this gut bacteria can be improved in disease, so that the disease also can be improved. Okay, can be like in diabetes, if they can be more under control of sugar and so on. Okay, so that's something about microbiome study. Okay, and that was my last slide.